is Kaushika Patel, Assistant Professor, Department of Pharmaceutical Technology. I welcome you to the MOOC series, an initiative started by LJ Institute of Pharmacy, Ahmedabad. In this session, we are going to discuss about the growth and the cultivation of bacteria. So let's begin with the session. Now, in normal terms, size is an important parameter that indicates the growth. But bacteria, they divide very fast by binary fission and there is an exponential increase in the number. So, magnitude of growth is expressed in terms of cell number rather than cell size. Okay, so growth curve of bacteria, when we want to describe, we have to plot a graph of cell number versus time, and different phases of growth can be shown in that graph. Okay, so here you can see the growth curve of bacteria. The number of cells, they are plotted against time. Okay, zero time indicates the time of the inoculation of bacteria on the growth medium. Okay, so four different phases are observed. First is the lag phase. Okay, where you can see that there is no increase in the number of bacteria. Why? Because during that phase, the cells are getting prepared for the division, they are absorbing the nutrients and they are getting ready for the process. So still in that phase, there is no increase in the number of cells. Okay, that is the lag phase. Second is the exponential phase, or it is also known as log phase, because here the number of bacteria will increase rapidly in an exponential manner. There will be a visible and notable increase in the number of bacterial cells. Okay, so you can see that there is a great increase in the curve pattern, and that is known as exponential phase. Okay, then will come the third phase, that is the stationary phase. Again here the number of cell increase has stopped. Okay, a plateau phase has been achieved. Why? Because the number of cells which are getting generated, on the other hand, same number of cells are getting died. Okay, the nutrients which are present in the culture media, they are getting depleted gradually, and that's why whatever is the increase in the cell number, there is same on the other side, there is decrease in the cell number. So a stationary phase is observed. Okay, and then finally, when all the nutrients are depleted, too many toxins have been accumulated in the growth medium, afterwards there will be the decline. Okay, decline in the curve will be observed, that is the death phase or decline phase, where there is decrease in the number of cells. Okay, this phase is observed if you continue the incubation period even after the stationary phase has been achieved. Okay, so growth curve describes four different phases of bacteria. Now, when you want to cultivate the bacteria artificially in the lab, what is required? You will require a proper culture medium containing all the nutrients. Okay, so nutritional requirement should be fulfilled. Then, when you provide the incubation, proper environmental conditions should be provided for the growth of bacteria. That importantly includes temperature, pH, and oxygen. Okay, so based on the important requirements, you can classify the bacteria or you can give proper appropriate name of the bacteria. Okay, so based on energy source, you can say that there are two types of bacteria, phototrophic and chemotrophic. Phototrophs, they will gain the light energy from the light, whereas chemotrophs, they gain the energy from the chemical compounds. Then, based on energy source, you can see that the bacteria can be organotroph or they can be lithotroph. Okay, organotrophs, they will use organic compounds as the electron donor, whereas lithotrophs, they use reduced inorganic compounds as electron donors. Okay, now you can further classify them based on energy requirement. They can be photoorganotrophs, chemoorganotrophs, photolithotrophs, or chemolithotrophs. Then based on carbon source, you can see that the bacteria, they are autotrophs or heterotrophs. Okay, autotrophs, they synthesize their own food, whereas heterotrophs, they utilize the energy from the organic substances. Okay, if living source is used as a food, they are parasites. If dead bodies are used as a source of food, then they are saprophytes. Okay, again, based on energy requirement, they can be classified as photoautotrophs, photoheterotrophs, or chemoautotrophs or chemoheterotrophs. Okay. Now, based on temperature, bacteria can be of three types. Okay, they can be psychrophiles, mesophiles, or thermophiles. Psychro means cold. So psychrophiles, they grow in cold environment. Okay, that is 0 to 15 degrees Celsius. Mesophiles, they grow in the moderate temperature. Okay, around room temperature 25 to 40 degrees Celsius. And thermophiles, they grow at high temperature, that is greater than 45 degrees Celsius. Now, in each category, you have obligate and you have facultative. When obligate bacteria are there, they are very stringent and they grow only in that specific conditions. When you have facultative, flexibility is allowed. Plus or minus condition is if it's possible. If it's provided, they can survive. They will not get killed. Okay. So obligate psychrophiles, they grow between 0 to 15 degrees Celsius, but facultative, they can survive up to 20 to 30 degrees Celsius temperature. Thermophiles, if obligate are there, they grow between 45 to 65 degrees Celsius. Facultative, they will grow between 37 to 55 degrees Celsius. Okay, but extreme thermophiles are available, which can survive at a temperature up to 110 degrees Celsius. Then based on pH, you can see there are acidophiles that grow in acidic pH, neutrophiles that grow in neutral pH, and alkaliphiles that grow in basic pH. So accordingly, the pH of the medium should be adjusted. Then based on oxygen, okay, you can see that bacteria can be obligate aerobes, obligate anaerobes, facultative anaerobes, or microaerophiles. Okay. Now, obligate aerobes, okay, they are very strict in oxygen requirement. They can survive only in the presence of oxygen and they grow on the surface of the test tube. Obligate anaerobes, they are strictly anaerobic organisms. They can survive only in the absence of oxygen. Even if small amount of oxygen is present, it acts as poison. Okay, so they grow at the bottom of the test tube. Facultative anaerobes, okay, for them the anaerobic condition is preferred, but even if little amount of oxygen is available, they can survive. That's why they grow throughout the test tube. Microaerophiles, they grow in a very lower concentration of oxygen only. They cannot survive higher amount of oxygen. Okay, so they also grow. They grow somewhat below the surface of the test tube. And aero tolerance, they can again grow in both the conditions. Now, so if you are asked what are the factors which affect the growth of bacteria, okay, then you can have chemical factors and physical factors. Chemical factors will include all the different nutritional requirements, which are nutritional components which are required for the growth of bacteria, like carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. All this should be provided. Along with this main component, they also require trace elements in very minute quantity. Okay, they also require growth factors like amino acids, purines, pyrimidines, and vitamins for the synthesis of nucleic acid for nuclear material. Okay. Physical factors, they include the parameters. Okay, we have already discussed temperature. They have to provide proper temperature depending upon their nature, pH. Osmotic pressure is also important. You have to provide a proper osmotic and pressure, okay, that is adjusted by 0.5 percentage sodium chloride in the culture medium. Light, okay, if bacteria require light for growth, then light should be provided. Otherwise, the culture medium should be kept in the dark environment. So culture medium, what is culture medium? It is artificially prepared mixture of all the nutrients for the growth and the multiplication of the microorganisms. Okay, they are also useful for isolation, purification, transport, and identification of the bacteria. Okay, so when you are preparing the culture medium, you can add different components to provide the different nutrients. Okay, so you can add peptones for as a protein source, beef extract for the source of nitrogen and carbon. You can use yeast extract for the vitamin source. You have to add sodium chloride as an osmotic pressure adjusting agent. Okay, and you have to add solidifying agent in case of solid culture medium. Okay, so now different types of culture medium are possible, okay, are available. You have to select a proper one depending upon your requirement. Okay, so three types of classifications are possible based on physical state, based on chemical composition, and based on their application. Okay, so based on physical state, there can be solid medium, liquid medium, and semi-solid medium. Solid medium, they are solidified in a Petri plate, okay, by using a proper solidifying agent like agar or gelatin. Liquid medium, that is known as broth, okay, that is available in the liquid form, that is used in test tube. And the semi-solid will have consistency between solid and liquid, okay, and they are used for special purposes. Nutritional requirement, okay, based on nutritional component, okay, there can be simple medium, there can be complex medium, there can be synthetic medium. Simple medium, that is the basic medium, which have all the components for the growth of all the different microorganisms, like nutrient agar or nutrient broth. Complex medium is the one that is obtained, that is having some components from the natural source whose exact composition is not known. So it is also known as undefined medium or natural medium. Like blood agar, that will contain blood. Now you don't know what is the exact composition of that blood. Synthetic medium is a medium which is prepared artificially, synthetically in the lab, whose exact formula and components you know along with their quantity. So that is a known or defined medium, like peptone water. Okay. Now, based on function, there can be different types of culture medium. So, first is selective medium. Selective medium, as the name suggests, allows only a selective species of microorganisms to grow on it. It will contain an inhibitory substance that prevent the growth of all the other species. Okay, so for isolation of the specific species, you have to use selective medium. Then there can be differential medium, okay, that is used to differentiate between two groups of microorganisms on the same culture medium based on their cultural and morphological characteristics. Okay, it does not contain any inhibitory substance. Based on the cultural characteristics, you can differentiate the two different groups. Okay, now if you see the example, then McConkey agar, that is both selective and differential. It is selective because it allows only the gram-negative organisms to grow on it. Gram-positive organisms, they are inhibited. And in that gram-negative organisms also, you can differentiate between lactose fermenters and lactose non-fermenters. Lactose fermenters like E. coli, they give pink colonies, whereas lactose non-fermenters, they will give you colorless colony. Okay? Then there will be enriched medium. Okay, as the name suggests, it will be enriched by some growth promoting components like blood, serum, or yolk. Okay, it is normally present in the form of solid culture medium. Okay, you have to use a Petri plate, and that a particular species growth will be more. It does not contain any inhibitory substance, but there is enrichment medium. This enrichment medium, okay, that also increases the relative number of a particular species of microorganisms, but it can have inhibitory effect or it can have growth promoting effect. Okay, it may increase the growth of a particular organism or it may inhibit the growth of other organisms, so that particular organism is more in number in that culture medium. This enrichment medium is normally in the liquid form, in the form of broth. Okay. Then there will be transport medium. Okay, it is used to carry organisms from one place to another, especially for clinical diagnosis. Okay, for clinical diagnosis, up when you carry the organism sample up to the clinical lab, up till there the organ bacteria can survive in that medium. For example, starch medium. Okay, they will preserve Neisseria and Trichomonas vaginalis in the cerebrospinal fluid sample. Then there will be anaerobic media. Okay, as the name suggests, they help or they allow to grow anaerobic bacteria. Okay, they will provide the condition where oxygen is absent. Okay, they will provide reducing environment and allow the growth of anaerobic bacteria like thioglycolate fluid medium or Robertson cooked meat medium. Questions are asked from it. So these are the frequently asked questions. Okay, thank you for watching the video. Please, for more updates, like, share, and subscribe our YouTube channel Pharma Ignite. Press the bell icon for the notifications. Thank you and see you soon.